Hello, everyone, and welcome to today's live broadcast, Analytical Method to Measure Empty and Full AAV Particles, presented by Christine Lebecq, PhD, Head of Analytical Development, Genethon. We are excited to bring you this educational webcast presented by LabRoots and sponsored by Beckman Coulter Life Sciences. Beckman Coulter Life Sciences delivers innovative and trusted scientific solutions across the globe for laboratory customers in a wide variety of settings: universities, government, biotechnology and pharmaceutical companies, hospitals, and commercial laboratories. For more information about our sponsor, please visit beckman.com/home. I'm Julie Simmeroff of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Now I'd like to introduce Dr. Christine Lebecq. Dr. Lebecq joined Genethon in 1997 and currently heads the Analytical Development Department. She is responsible for developing bioassays and setting up advanced analytical tools used for characterization and in-process testing of gene therapy products at early stage development. For Dr. Lebecq's full biography, please visit labroots.com. Please join me in welcoming Dr. Christine Lebecq. I will now turn the presentation over to her.
Thank you, Julie, for this introduction and for giving the opportunity to present our development regarding the analytical ultracentrifugation technology for AAV vectors. So first of all, I would like just to show you all the different parts of the presentation. I will make an introduction of Genethon. After that, I will introduce the process manufacturing and the quality control of gene therapy product, and especially for AAV vectors. I will make, after that, present the principle of analytical ultracentrifugation and the proof of the concept and the qualification of the method for characterization of empty and full AAV particles. And in the last part, I will present some application of for AAV particles. So first of all, Genethon has been created in 1990 by the French Association of Muscular Dystrophy. It's formed by the AFM, this French association. Genethon is an integrated R&D center. It's a non-profit organization. We are currently approximately 200 people, a researcher, technician, engineer, administrative persons. Our mission is to design and develop gene therapy products for rare disease. We are ensuring the translational development from research up to clinical validation. Genethon is developing therapy for rare genetic disease, neuromuscular disorder, immunodeficient eye and liver disease. Our platform is based on AAV and lentivirus vector. We are currently sponsoring several gene therapy projects that have reached clinical trial, and we are also involved in several other projects for AAV and lentivirus vectors in preclinical and research stage. For neuromuscular and liver and eyes, the gene therapy product is based on the AAV vectors. I will present you the different process we have been developed and used in at Genethon. First, it's the most common production process. It's based on one of the most common process is for production of small lots, is to transfect adherent cell line, human cell line with 3 different plasmids, one for coding for the transgene. the two other for the rep and cap, and for the other one for the adenovirus auxiliary function. This process is commonly used for producing AAV at a research grade, but in some cases for GMP lots at a small scale, because you will get like 24, you can make like this scale at 24 cell factory, giving you around 25 liters of bulk product. So this process has limited capacity, is flexible, fast implements, but limited capacity, and the cost is elevated per lot. So for that, we have another process in parallel, which is based, instead of using adherent human cells, we use suspensions human cell line, and the transfection is done in bioreactor, and the current cell is 200 liter in disposable bioreactor. So this process can give you more product, but it has also limited capacity to 200 liters, and it costs also a lot per batches, because you need to use plasmid GMP source. And for a large-scale production, instead of using this process, we use another process, which is based on the Baculovirus process, and we use a dual Baculovirus expression system. And you need for that one Baculovirus coding for the rep and cap of your gene necessary for making the AAV vector, and the other one is coding for the transgene. You infect insect cells, SF9, in the bioreactors. And the current scale is 200 liters in disposable bioreactors. So this process has a capability to be scalable. You need to, the implementation is slow because you need to generate the Baculovirus bank, but the cost is smaller compared to the other process if you need to make high number of lots of AAV vectors. After that, I will present just the four on this slide, the downstream process for purification of AAV. So the it's a four different measure steps. So from the harvest, you make a clarification of the harvest. You purify the AAV vectors on human chromatography, and you make after that an ultrafiltration, concentration, and change and make your AAV in the formulation buffer just to obtain the drug substance. And in the last step, you make a sterile filtration, and you make the fill and finish to obtain the drug product. The AAV production at Genethon is based on these three different processes: in transfection in cell factory, in transfection in bioreactor in suspension, or using the Baculovirus system. We use this different process depending, of course, of the yield of the process, depending also of the need of the vector for making your clinical trial, and for the need of the vector for the administration to the patient. So this process is used for AAV2, AAV8, and AAV9 serotype, and is for different clinical projects. After that, after the production, the quality of the product will be evaluated, and the design of the quality control package is based on the guidance on assay required for product release testing and for the characterization of the product. The quality of the AAV product can be divided in three different groups. The first one is the strength and the dose, and it's the viral genome titer, the VG titer, the infectious titer, the activity and the potency of your vectors. The second one is making the characterization, the identity of the vectors, in terms of the DNA genome, in terms of the viral particles, in terms of your buffer, pH osmo, and the ratio of full and empty capsid. So the third group is for the purity. You can need some assay to quantify the residual DNA, the residual proteins, the residual chemical agents. It gives you the monitoring of your vector's purity. And the last one is the safety of your making the sterility, advantage the safety of your product. In terms of ratio and of full and empty capsid, you will have, of course, in the AAV productions, in the synthesis, you have different type of population. The full particles, meaning that you have in your particles the full vector genome of your interest. The partially full, it's AAV particles that have encapsulated nucleic acid, smaller than the genome of the AAV, but also can have some particles, some DNA coming from the process, like as the DNA, the cellular DNA, the plasmid DNA, the bacular DNA, and the empty particles does not have any nucleic acid at all. So you have different analytical assays to quantify AAV particles, to quantify the full particles, and to quantify the total particles, the mix of empty and full particles. To quantify the full particles, the method usually commonly used is a qPCR assay, and gives you a concentration of your viral genome. Does not have any limitations. The ELISA, it's an ELISA kit which is commercially available. It gives you a quantification of the viral capsids, but this kit is not available for all serotypes, and he has this limitation. And but if you have the kit available for your serotype, you can make a quantification and the ratio of the full and empty by making the difference between the ELISA and the qPCR methods. You can also make a measurement of your quantification of the total particles by the SDS-PAGE. For that, you will need a high quality of a high purity of your AAV product, but you will need also a reference standard material just to make a standard curve and to make a quantification relation between your product and the reference standard. Some people make a quantification by spectrophotometry at 260 and 280 and give you a relationship between the protein and the DNA contents. And for this method, you also need the reference standard material. By HPLC, you can make a separation of full and empty capsids. This has been this has been published by other lab and for AAV2 and AAV8. But this is serotype dependent. This is transgene dependent, and give you a low resolution. So you can have from this method not a separation, but mostly a quantification of the total particles. The nanoparticle tracking, namely NTR or the nanocytes, it's a quantification of viral particle, but since the AAV particle it's small, it's only 30 nanometers as a diameter, you need to label the AAV particles before to make a quantification. The electron macroscopy give you an observation of the viral particle as a population, and you can make a quantification of the full and empty particle. But this technique is difficult to distinguish the partially full to the empty and full particles. So our challenge was to develop one method which is independent of the serotype, independent of the transgene, independent of the process, and can give you the ratio of the full and empty particles. And we tried at the beginning a few years ago, almost 10 years ago, just to use analytical ultracentrifugation to make a quantification of the full and empty particle. And at the beginning, we are basing on what has been published in 1980. we know that AAV, you have the heavy and light particles for full and empty. And in these publications, they present the full particles, which has an S value around 110, and the empty particles at different density, and the S value is around 66 S. So from this paper, we try to make to apply the AUC just to have an AAV profile, a profile of the full and empty particles. So from that, go back first to the principle of the analytical ultracentrifugation. The AUC, it's a technical tool. We give you actually a very good real-time collection of a signal data by absorbance and Rayleigh interference systems, and you can follow up the sedimentation of your profile during over the times. And this you can follow up also by absorbance optical at the wavelength to 200 until 800 nanometers, or by Rayleigh interference. And we try, you can see, you can have either a rotor on the right panel, you can have either a rotor of 8 samples or 4 samples. So you can run independently 4 or 8 samples depending on the rotor. We try to apply, so we apply the sedimentation velocity application just to make a separation of full and empty particles. So from that, at the beginning, what you have in your cells, you have, at the beginning of the run, you have an homogeneous signal in the meniscus until the bottom. And when you run the sample, when you start the sedimentation, you try to run, and you will form a boundary around over the times. So this boundary will give you a difference in terms of OD, and you will see a difference between the meniscus and the bottom. You will see the boundary, and you can follow this boundary over the times, either by OD or interference. And this boundary motion is dependent only on the molecular weight and the molecular shape of your product. You don't need any standard molecule for calibration. This calibration is only based on the distance, the times, and the temperatures. The data scans you will have, like on the right panel, you can see that you will have over the times the scan, and the data scan can be transformed after, like a distribution profile. And for that, you use the SetFit software just to make a profile of your data. The optical system is two different, one by UV and the other by interference. And UV use the scanning speed, it's 1.5 minutes per wavelength per cell. You collect at two different wavelengths. And for interference, this is faster. The scan of the cell over between the meniscus and the bottom is 10 seconds per cell. The sensitivity, it depends, of course, of your product. You can choose a wavelength depending on your product, the absorbance. And for interference, it does not have any selectivity, but you have also a high sensitivity of the signal. So for the wavelength, in case of the AAV, you will need, of course, you will choose where you will have a flat signal, so between 260 and 270 nanometers. And in this range, the signal should be between 0.1 OD and 1.5 OD. This is the dynamic range of the absorbance signal. For the interference, the dynamic range is unlimited. You have very wide range until 0.5 milligram per ml to 2 milligram per ml of protein. The protein needs to be at a minimum in the research grade, and for interference you need a high quality, and it's very dependent of your buffer. So first of all, I would like to show you some experiments we have been performed here at Genethon just to make a qualification of the method. So for that, we start at the beginning with a process with a lot, which an AAV preparation, which has a mixture of full and empty particles, and we make the separation of the full and empty particles by cesium chloride reagent. And we collect from the syringe, you can see in the middle of the slide, the full particle to the empty particles. And after that, we make analytical ultracentrifugation analysis of different preparation. So before, I told you previously that the motion and the boundary motion will depend of the shape of your product and of the molecular weight of the product. And for that, we also control that we do not have any difference in the shape, in terms of shape, between the full and empty particles. And we make, first of all, the microscopy analysis just to see we do not have any difference. And we also check by dynamic line scattering, and we verify that the size of the AAV particles, it's almost the same. And as you can see here, in red, this is the empty particles, and in blue, this is only full particles. And the diameters of these empty and full particles are consistent and almost the same things, the same size, around 26 nanometers. So after that, we run and we analyze the empty particles. In blue on the left panel, this is the AAV empty particles. We have a nice peak at 60. On the right panel in green, this is the AAV night empty particles. We have also one single peak, around 60. When we overlay, we can see a very nice overlay of the AAV8 and AAV9, meaning that the S particle is around 61, 64 S. And the methodology, the AUC, is serotype independent. We also run analysis of several lots at different stages just to see the repeatability of this method. And for that, here you can see the overlay of 5 different prods, 5 different preparations. And the S value is around 63 to 65, which is almost the same things. It's very similar. And we have a constant value. We run this for 10 independent experiments. We have an average of the S value around 64, more or less 1 S. So we have a very nice repeatability of the experiments. We also look of the linear range of the methods, and for that we took the one batches and we run the experiment with that and dilute in purple, dilute by half and dilute by 4, and we can see that the S value is almost the same, and it's around 62 to 66. And we have, and you can look at the area of the peak is proportional to the concentration of your product, meaning that we have a very right linear range, linearity of the signal between 0.2 to 1.2 OD. After that, to make a characterization of the empty particle, we look at the full particle. For that, we take one example, the AAV GFP. This lot has been produced by triple transfection in human cell, purified by cesium chloride, and we make a quality control on this product, and we make a titration of the VG titer, and we make the total particle, and we have a very nice correlation between these two assays, meaning that we have only full particles. When we run the AUC, you can see one peak around 102 S, and by UV or interference, we have the same signal, only one peak, meaning that we have the S value is dependent, of course, of the mass of the DNA. On this slide, I show you the overlay of the empty particle, AV8 in blue, AV9 in green, and AVGFP in purple. The S value for the empty particles is around 64, and the full particles, it's around 70 to 110 S, depending on the mass of the encapsulated DNA. If we go to the theoretical consideration and to go back to the Svenberg equation, what does it mean, the S value? The S value is dependent of the mass of your particles, is dependent of the V bar of your particles, and of the friction, the coefficient friction of your product. So the S value of the empty particles is only depending of the mass of the capsid and the V bar of the capsid. The S value of the full particle is related to the mass of the full particle, so of the mass of the capsid and the mass of the DNA. So if you combine these two relations, you can see that the S value, the S value of the full particle is correlated to the AAV vector genome size, to the mass of the DNA. So for that, this is the theoretical, and we demonstrated this by running different products, different prep, which has different size of encapsulated DNA. So on this slide, we can show you the sedimentation coefficient in function of the size of the DNA for AV2, AV8, and AV9 in blue, red, and green. You can see if you do not have any DNA size, for all you have the S value around 64, and the S value is increasing and linear increasing until 105 for an encapsulated DNA of 480 nucleotide, which is the maximal size of the DNA inside the AAV particles. So the S value is proportional to the size of the transgene, and you can make a very nice analysis of your particles. So in summary, we can have a characterization of antiparticles, full particles, partially full particles, which mean intermediate form of AAV, depending, of course, of your size of the transgene. You can also show some free viral particles below 60, and you can see maybe some aggregate if you make some dimer, something like this, over 120 S. So, the main advantage of this method, it's a matrixing, it's independent of the buffer formulation, it's independent of the serotype, independent of the transgene. You can run any different product, difference in one assay. And the limitation needs, you need at least a high quality of purity is required. You need at a minimum 10 to the 11 Vg per ml, and the volume required for that is 400 microliters. And the maximum volume is 400 microliters. The minimum is 100 microliters. Now I will show you some application of the AUC. We have used this AUC technology just to analyze self-complementary AAV vectors versus single-strand AAV vectors. Self-complementary, it's some DNA, some AAV particle which has encapsulated a double-strand DNA instead of single-strand DNA. So we have made some double-strand AAV particles, GFP particles, AAV GFP, and we have on the left panel, you can see we make a characterization of the DNA, And you can see by alkaline gems that we have only one band at 4,600 base. And on the right panel, we have made this AAV analysis by AUC, and you can see one peaks for 3 different batches. We have mostly one peak around 64. This is empty particles, which represent around 86%. And we have at 103S the full particles, the double-strand AAV particles, which represent around 7%. For another project, for AAV night, we compare AAV night production to the double strand to the single strand. So on the SDS-PAGE, for the viral particle, we have the same profile in terms of the VP proteins, in terms of the ratio of the empty and the full particles. You can see for the double-strand AAV, you have almost 90% of empty particles. For single-strand AAV, you have 22% of full particles, meaning that you have encapsulated, the encapsulation is lower with the self-complementary compared to the single-strand. So it gives you, the AUC gives you an idea of the full and the ratio of the full and empty, even for the single-strand and double-strand AAV vectors. We also use this machine, the AUC technology, to compare the AAV product, AAV vector produced either by a Baculovirus system or triple transduction. So on this, as I show you, we have 2 different processes in bioreactor, and before to choose between the Baculovirus system to the transduction system and to make the production at 200 liters, we use these 2 different processes at a very small scale, at 2 liters and 10 liters, and we look the quality of the product and the yield of the process. The quality of the process, so here you can see the SDS-PAGE, and on lane 1 and 2 it's for the baculo process, on lane 3 and 4 is the transduction process, and you have almost relatively the same purity in both processes. In the Baculovirus process, you can see some extra band above VP3 due to the cleavage of VP1 and VP2, but the quality is almost the same. We look after the ratio of empty and full capsid by AUC. For 3 different batches of 2 liters, you can see you have around 27 to 31% of full particles. For the transduction process, we are around 16 to 18 of full particles. And we have almost the same process at 2 liters and 10 liters, and we have the same amount of full capsid in both processes. However, we have up to 20% of intermediate species for the Baculovirus process compared to the transduction process. So when we obtain that profile, we look for to understand and to make a characterization of what was the intermediate species. So for that, we make a fractionation by ultracentrifugation on cesium chloride, and we collect all the different fractions from the bottom of the tubes, and we make some fraction, very small fraction around. We make from that around 16 fractions. And from all collected fraction, we have performed the SDS-PAGE analysis, the qPCR assay, and the AUC analysis of selected fraction. On this slide, you can see the Capsids distribution. On the left panel is for the Baculovirus. On the right panel is a transduction process. And from all the fractions from the one, it's from the bottom of the tube, which is empty. And from the 17, which is on the top, for the heavy particles. So you can see the profile on the graph. It's a distribution profile for the VP protein over the fraction. And you can see that the fractionation confirms the AUC profile that the baculovirus system generates abundant capsid of intermediate density compared to the transfection process. We have done from the fraction 5 and 14 for the baculo, and from the fraction 8 to 13. So here, this is empty particles. You can see the AAV vector distribution. So here on this slide, we make the we analyze the distribution of the vector for So on this slide, we make an analysis of all the different, not all the different fractions, but the fraction which contains some VP particles, fraction L until 13, and you can see the full particles, which for AV8, fraction 8 and 9. You can have a nice peak around 100th. And the full, the empty particle, which is in red and yellow, it's for fraction 12 and 13, and we have this the intermediate spicy in the light blue. For the baculovirus system, we have more fractions, and you can see the S value. It's for the fraction 5 and 6. We have an S value around 100th. All the intermediate spicy are included between 70th and 19, 5th, and the empty particles, it's around 64, 65th. So the baculovirus system generates abundant capsid of intermediate density. We have also analyzed by qPCR by targeting some sequence of the transgene of the genome DNA, just to analyze what is the intermediate spicy, and in blue by targeting the RTR sequence, and in red by targeting a specific sequence in the transgene. And for the baculovirus, you can see on the left panel that you can see you have some transgene, some specific full particles in the fraction 6, but you have also some fragmented DNA vectors in the fraction 7 to 11. And for the transfection process, you do not have this fragmentation of the genome. We also look for all the contaminants coming from the process, in green from the baculo and the plasmid contents, and in purple for the cellular DNA. And you can see on the graph that almost this impurity are present in the fragment as a small part, a small fragment, small size, and in the fraction 7 to 12 as intermediate spicy, and also the intermediate spicy for the transfection process contain only this type of contaminants. We have to notice that this specific contaminant, this non-specific DNA, represents less than 0.1% of specific vector genome, which is a very small amount. So the AUC method can give you the profile of the different process, and give you for the baculovirus, we have the same amount of the full particles, around 27, but we can see by the AUC that we have more intermediate spicy for the baculovirus compared to the transfection process. So the AUC testing allows to select the production process. In the last slide, I would like to show you the control. We use this technique just to control the manufacturing process productions. And we have few examples. We make this for some AAV2 productions in adherent cells, human cell line. And on the left panel, you can see the SDS-PAGE, the VP profile, and on the bottom, you can see the AUC profile of this of one of the batches. From these 9 different batches, the S value of the full particles is around 96S, more or less 1S, and represent around 67%, more or less as 8%. So the AUC technology method is applied to ensure the lot-to-lot consistency in our case. We also check for the vector can inform you about the quality of the product during the scale-up of the process. And you can see on this slide that we have run some different batches, the same product, but at different scales, 50 liters, 150 liters, and 200 liters. And the profile, the AUC gives you the profile of your vectors, and it can give you an information about the manufacturing process when you scale up the process. And the last example is for optimization of the manufacturing process when you want to compare to one process to another one by changing side by side or one step. In our case, I can show you here when we have one process, we have around in black 20, around between 10 to 20% of full particles. You change only one parameter of your process and you can increase until 40 to 50%. We use this for different type of changing one process. So the AUC can give you a monitoring of the product enrichment during the optimization of the process state. So you can choose in that case the process even from the yield and also for the ratio of full and empty particles and the percent of intermediate spicy. In conclusion, the AUC, it's a complex machine. It's some kind of expensive. It's more than 400,000 euros. But the equipment and data analysis are not automated. It requires an extensive and proper training. But the AUC is a potent tool to analyze the AAV product profile in the final product. You can make an identification and a quantification full, empty, and other particles. You can make the characterization of subspecies in the preparation with different molecular weight. You can also compare the self-complementary genome to the single-strand genome. And you can use this as a routine testing to inform the product development process and to demonstrate the lot-to-lot consistency. In the last slide, I would like to acknowledge all of Genethon, my group, the analytical developments, and especially Florence, Cyril, and Lou, who run all the analytical analysis, the process development, and especially Matthias Eben, who provide all the different batches, Otto, Mehdi, and Federico for all the different interactions we have, and Bertrand, especially Bertrand Renal from Institut Butter, which we start to make the proof of the concept with them. Thank you very much for your attention.
Thank you, Dr. Lebec, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the Send button. We'll answer as many of your questions as we have time for. If we are unable to get to your question, Dr. Lebec will follow up with you via email after the presentation. Let's get started. Our first question is, did you use some orthogonal methods like those described in your presentation to quantify the ratio of full and empty particles? If yes, did the quantification correlate with the results obtained by AUC?
Thank you for this question. Yes, we use some different methods that are presented in the table. And we use the ELISA kit for AAV8 particles, and we have already a nice correlation between what we obtain by AUC and what we obtain between the ratio of the ELISA and the qPCR method. But as I said, the ELISA is not adaptable for all of the different serotypes. We also have a nice correlation from cryo-TEM by electron microscopy, but this technique can give you a sub-estimation of the full particle, as you cannot compare. It's very difficult to distinguish between the partially full and the full to the empty particles. So usually the partial full is usually a count like a full particles, and you underestimate the percent of the full particles.
Thank you. You perform AUC technology as a routine test to monitor the quality of the AAV vector. Do you plan to validate this method for a quality control of cGMP lots as a release testing?
Right now, we make some qualification of the method. As I present, we make some repeatability just to demonstrate the repeatability and the linear range of the signal. For making a full validation of this method for quality control of cGMP lot as a release testing, we should validate the software. And so far, it's really difficult. The software has been developed by an academic university, and we don't plan to use this as for release testing of GMP lots. But I heard, and some people try to do this, actually.
Thank you. The next question is, are you using the AUC as an indicating assay in the stability studies?
Yes, we use this AUC for stability study for showing if we have any different form during stress conditions. If you make a freeze and thaw samples at a different time, like 3 to 5 times, or if you leave your sample for 1 month at room temperature. So far, we didn't see any degradation of the AAV particles. We didn't see any difference. But Dimensions, the societies by some authors use also this technology just to as an indicated, and it shows some different form at 37 degrees.
Great, thank you. We'll wrap up with one last question, and that question is, how long does it take to run one assay?
Actually, we have here at Genethon the Beckman XL1, and for the AAV, we run at only one wavelength, and we run at the same time the interference. And we only can run 3 samples in one run, because if you run 7 samples, you lose some scanning. You lose some scanning intensity data, and you cannot record sensitivity and everything. So it's not so good to run more. We can only run 3 samples in one day.
I would like to once again thank Dr. Lebec for her presentation. Do you have any final comments?
No, for me,
Okay. Thank you once again, Dr. Lebec. I would also like to thank LabRoots and Beckman Coulter Life Sciences for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 28, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.